right, we're going to go through the steps of cellular respiration. So if you look on Canvas, you'll be able to see this worksheet that has a way to organize your notes. This worksheet actually has the same exact information as this visual picture right here. It's just a way of organizing the steps and stages of cellular respiration. I think it's a useful tool. There's no question on the exam where I'll give you this exact chart and have you fill it out, but there will be questions on the exam that exactly relate to this chart. For example, where does the citric acid cycle happen? Or how many ATPs are produced by glycolysis? Or what are the products of pyruvate oxidation? So I think this is a great way to organize your notes. So we're going to start with glycolysis. As we go through each of these steps, we want to be thinking about where do each of the steps happen? What are the starting materials or the reactants or the substrates of that process? What are the products at the end? And by products, I really mean the carbon-containing products. And also, how much ATP do we make and how is it made and what electron carriers are made. I want to point out that I have organized this chart a little bit differently than one of the homework problems. In the homework, it's going to say that ADP and PI are a reactant, and the electron carriers, NAD+, is going to be a reactant, but I have just not included those in the reactants because our chart's just going to get too muddied up. So we're going to start with glycolysis. I want to point out that there's two different ways that ATP can be synthesized. Most of the ATP that our cells produce happens during the very last steps of cellular respiration called oxidative phosphorylation. But along the way, we're going to make a little bit of ATP by a process called substrate level phosphorylation. Now your textbook goes into the details of what are the substrates, what's the name of this substrate, what's the name of this enzyme that catalyzes the passage of the phosphate group from the substrate to ADP to form ATP. You don't need to know all of those details for this class, but I do want you to know which steps produce ATP by substrate level phosphorylation versus oxidative phosphorylation. And for glycolysis, we're going to use substrate level phosphorylation. So glycolysis is happening in the cytoplasm. So glycolysis, importantly, can happen with or without oxygen. So it can happen aerobically or anaerobically. This step is not directly dependent on oxygen. Glycolysis means glucolysis. I really like how your textbook uses these ball and stick models that just show the carbon. So this is a 6-carbon glucose molecule, okay? And what happens during glycolysis is glucose gets lysed. Glucose gets split in half. So for our reactants, our starting material is glucose, C6H12O6. I want you to keep track of the carbons. So we're starting with 6 carbons, okay? And what we're going to see at the end is the product that we make is called pyruvate. Each pyruvate has 3 carbons, and our glucose is effectively split in half to make 2 pyruvate molecules. Okay? So we have our 6-carbon glucose molecule as a reactant, and it gets split in half to form 2 3-carbon molecules called pyruvate. So this is an overall summary of the process, but there's actually a couple steps in between. To get glycolysis started, our cells actually have to use some energy to start this process. So we actually have to burn 2 ATP to start glycolysis. This step is called the energy investment stage. But in subsequent steps, we actually make ATP. For each round of glycolysis, 4 ATP are produced during the energy payoff stages. So overall, from glycolysis alone, we end up netting 2 ATPs. And the way that these ATPs were made in the energy payoff stage are through substrate level phosphorylation. So we made a tiny bit of ATP during glycolysis, but another important thing that happens is that the electrons are being transferred from glucose to our electron carriers. So glucose is becoming oxidized, and our electron carriers are becoming reduced. So the electrons are going from glucose to NAD+. And we actually end up making 2 reduced electron carriers from glycolysis. So we make 2 NADH. Next step is called pyruvate oxidation. And I call this kind of a half step because we actually don't make any ATP in this process. What happens during pyruvate oxidation is it brings the reactants of cellular respiration into the mitochondria. So where this is happening is across the mitochondrial membrane. The enzyme complex that does pyruvate oxidation is actually the target of arsenic poisoning. So pyruvate dehydrogenase is blocked by arsenic. And if you can't do pyruvate oxidation, your cells can't make enough ATP to survive. One thing to notice about our chart, the carbon-containing products of one step are going to be the reactants for the next step. So our reactants for pyruvate oxidation are pyruvate. I want you to remember that this step is going to happen twice for each glucose molecule because we have 2 pyruvates from our initial glucose. So pyruvate is the reactant. Each pyruvate enters pyruvate dehydrogenase and a couple things happen. Now remember, our pyruvates have 3 carbons each. The first thing that happens during pyruvate oxidation is 1 carbon dioxide is lost per pyruvate molecule. Another thing that happens is that electrons are stripped out. So NAD plus becomes reduced to form NADH. And then finally, the remaining 2 carbon molecule acetate is added to coenzyme A to re-energize it. And so that new molecule that's formed is called acetyl-CoA. Now remember, pyruvate oxidation happens twice for each initial glucose molecule. So from 1 pyruvate, we get 1 carbon dioxide, 1 acetyl-CoA, and 1 NADH. But because we have 2 pyruvates from each starting glucose molecule, we get total from 1 glucose molecule, 2 carbon dioxides, 2 acetyl-CoAs, and 2 NADH from pyruvate oxidation. So again, the function of pyruvate oxidation is to bring pyruvate into the mitochondria. So it's happening across the mitochondrial membrane, okay? And it also starts the breakdown of our carbon-containing products. So we start to strip carbons out of our glucose and make carbon dioxide. So don't forget the forest for the trees. Okay? The whole reason that we're doing all of these reactions is to get the electrons from glucose to oxygen. Okay? So in glycolysis, all we've done is start to break our glucose apart. One more thing to mention about pyruvate oxidation. The enzyme complex pyruvate dehydrogenase only functions when oxygen is present. So oxygen isn't a reactant, but oxygen must be present in order to do pyruvate oxidation. Without oxygen present, the pyruvate's actually going to stay in the cytoplasm, and we'll talk about what happens to the pyruvate in the absence of oxygen later. But I want you to note that pyruvate oxidation and all of the other subsequent steps are dependent on the presence of oxygen. So the next step that we're going to talk about is the citric acid cycle. Again, oxygen has to be present for this to happen, although oxygen is not a reactant. Okay? So where this is happening is in the mitochondrial matrix. So if we remember our structure of the mitochondria from chapter 5, we have an outer membrane, we have an inner membrane, there's a gap between those 2 layers. It's called the intermembrane space. Read carefully on your quiz questions and the homework because inner membrane and intermembrane space look very similar. And then the very inside of our mitochondria is called the mitochondrial matrix. Okay? And the citric acid cycle is happening in the mitochondrial matrix. All right, so just like in our previous steps, the products, the carbon-containing product of pyruvate oxidation is going to be the reactant for this step. So our reactant is acetyl-CoA. Acetyl-CoA has 2 carbons, and then the coenzyme A attached to it. Okay? But remember, for each one glucose molecule, this is going to happen twice, right? Because we had one glucose that got split in half to 2 pyruvates that each lost a carbon during pyruvate oxidation, and now we have 2 carbons left in each one in the acetyl-CoA. So what happens during the citric acid cycle is acetyl-CoA reacts with oxaloacetate. This is not a vocab word, you don't need to know it for the exam. But what happens in the process is the carbons get broken down and released as carbon dioxide. Electrons get stripped out and passed to our electron carriers, NADH and also FADH2, and ATP is made through substrate level phosphorylation. And in this process, oxaloacetate gets recreated, okay? So that's why it's a cycle, because when acetyl-CoA reacts with oxaloacetate, it goes through all of these steps and it makes oxaloacetate again. So when we think about what happens in this process, what happens during the citric acid cycle is we finish the breakdown of glucose into carbon dioxide. Each of those carbons that were in acetyl-CoA get released as carbon dioxide. But again, this happens twice, once for each acetyl-CoA. So I do this thing with my fingers, okay, to show you what's happening so far. We started with glucose. Glucose has 6 carbons. During glycolysis, we did glycolysis. We separated the carbons apart into 2 3-carbon molecules called pyruvate. During pyruvate oxidation, each pyruvate lost 1 carbon dioxide, and then during the citric acid cycle, the remaining carbons are lost as carbon dioxide. So during the first several steps of cellular respiration, we finish breaking down glucose and releasing carbon dioxide. So we get rid of glucose, we make the carbon dioxide, but so far we haven't used any oxygen and we haven't produced any water. That doesn't happen until the subsequent steps. So going back to our chart, our products are 2 carbon dioxides per acetyl-CoA, or 4 total per glucose we started with. Sorry, we're getting a little out of the way here. We do make ATP through substrate level phosphorylation. We make 1 ATP per turn of the cycle, and so we make 2 ATP per glucose molecule. And then we do make electron carriers. We actually make 3 NADH per turn of the cycle, and another reduced electron carrier, FADH2. But again, that's for each acetyl-CoA. For each initial glucose, we actually make twice as many of these. So you should be able to know and recognize that the citric acid cycle happens twice per initial glucose. So if I tell you the citric acid cycle makes 3 NADH per turn, how many NADH will we get from 5 glucose molecules? All right, well, let's think about that. 5 glucose molecules will make 10 pyruvates and 10 acetyl-CoAs, so that would make 30 NADH. Right, so as I said before, going through the first 3 steps of glycolysis, pyruvate oxidation, and the citric acid cycle, what we've accomplished is breaking down glucose and releasing carbon dioxide. But at this point, most of our energy is locked up in our reduced electron carriers, NADH and FADH2. And it's in the last steps that those electrons are going to get transferred from the reduced electron carriers to oxygen, and most of the energy is going to be released that produces most of the ATP that our cells need. So these last 2 steps, where the electron carriers become oxidized and pass their electrons to oxygen and oxygen becomes reduced, and the energy that's released is used to make lots of ATP, is called oxidative phosphorylation. And that's what we're going to talk about next.